chemicals, electrical messengers and chemical messengers that pass between, this is called a synapse, and we'll get to all those different parts of the neuron. And so some people like to say, oh, well, all of our, intelligence, our creativity, can we really strip it down just to chemicals and electric messengers? Can we get it? We can literally take it down to this very basic unit. And we'll talk about what those basic neurotransmitters are today, the most common ones that we talk about in psychology. So we've got a very basic concept of synapses, neural communication in the brain, electrical activity, chemical messengers, and it translates into very, very complex human behavior. Look at, you've got this, maybe this is a mother and her child, and you've got so many things that are going on here that are on a very complex level, right? We're seeing some kind of reciprocal behavior, right, between mother and child, emotion on their faces, and actually in the case of the child here, this is actually pre-wired in her brain. She's ready to receive information, and she will reciprocate when she sees those facial expressions. She will copy them. And so they've discovered this specialized neuron system in the brain called mirror neurons that probably there was a switch in evolution at a certain point in time that allowed us to succeed because we were able to have that next level of communication just through facial expression. You go, okay, that person is smiling, they're friendly, they're not going to try to hurt me, and you learn these things instantly. So the baby learns them instantly from watching her caregivers, and then she mimics it and copies it. And then the children are also pre-wired and ready to learn and engage in these social behaviors. So yes, non-human primates like chimpanzees and gorillas, and they are very social, and they do sort of chatter and communicate with each other, and a lot of animals live in groups, right, and sort of seem to socialize, but not on the same level as humans, right? Is this older boy helping the younger boy? He's teaching him somehow. Something's going on there that's social about their engagement. These two children, again, of course you've got these very basic needs of eating and getting enjoyment out of that. So the child gets some neurotransmitter released from their brain, the chemicals that say, ooh, I like this ice cream, it tastes good. And she's egging him on maybe for a bite. He gives her a bite. Is he thinking, these kids are maybe 3 years old, 2 or 3 years old, is he thinking, oh, well, tomorrow she might have an ice cream and I might want a bite, so I'm going to give her a bite now? He's probably not thinking that. He's not that conniving, right? He's not at that point yet. So what is it, about our brains that want to be social and emotional in our learning? This is a picture that obviously we as adults are just kind of reading into. We don't really know what these babies are feeling. Babies are born with basic emotions, and you typically don't see secondary emotions until the second year of life. But what emotion do you imagine, or do you infer when you look at this baby here on the right? What emotion might you call that? Empathy. Great. Yeah, empathy. She sees this other baby that's crying, and I don't know, maybe she's curious and going, what's going on here? But she's not crying, and doesn't have an angry face like, stop it, you're being too loud. She's got this sort of empathetic concern, and actually empathy is one of those emotions that we really don't see in any other species outside of humans. Some people like to think they see it in their dogs or in their chimpanzees, and maybe they're being trained to do things, but at what levels? Is it at the same level as humans? And empathy can be taught, so that's good news, right? But some kids are naturally more empathetic than others, and sometimes you see that sooner than psychologists used to think in the past. They used to think, oh, we wouldn't be able to see these kind of higher level emotions until 3 or 4 years old. But now we think maybe empathy can be present as young as 18 months. These kids are not probably about a year. So all of that complex human behavior and emotion, thought processes, can we strip it back down to neurons and chemical messengers? Well, that's our attempt today, just to get sort of the basics under your belt as to how this works. So we will talk about the anatomy of the neuron, so the basic structure and function. The major important neurotransmitters, now there are hundreds of neurotransmitters that are produced in your brain, but we have a few that we hear about over and over and over again, especially in psychology, because they often have to do with the reward system or consciousness or alerting your nervous system. And so that's what's important to behavior and emotion. Then we'll get into the nervous system. That might wait until our next class, and then around the major brain areas and understanding their functions. Yes, there is a lot of memorization this week. I really recommend that you get your flashcards going this week. Start doing that now, because then as we keep kind of gaining on our topics week to week, it will just be a knowledge base that you will know. You'll have that to fall back on. And by the time we get to the final exam during finals week, you'll have questions that, quite honestly, you may not know what part of the textbook it came from, because a lot of them are going to be about the nervous system, the brain, the neurotransmitters, because we revisit them with almost every topic. Okay. All right, let's ask a question here. Get out your clickers. Okay. How many neurons do we have? About how many neurons? We're on channel AA. Three, two, 1. Stop it there. See what people think. Okay, great. 50% of you did your reading so far, or at least have some of this basic knowledge. So about 100 billion. I read an article recently where the author said 97 billion, and I'm not sure where that number came from, but roughly 100 billion neurons, okay? A lot. They are densely packed in your cortex, the cortex of your brain, and about 2/3 of them are there on that cerebral cortex, and the other third are going to go down your spinal cord, and then all throughout your peripheral nervous system. Are we born with all our neurons? Okay, I'll stop it in 3, 2, 1. Okay. So the answer to this is yes. We are born with all our neurons. So the average baby in North America weighs about 7.5 pounds, 20 inches long. And so that's a pretty small skull, right? It's got to be, it has to be able to pass through the birth canal, which is dilates to 10 centimeters, which is 4 inches, which is a drastic change in the female body during labor and delivery. That is drastic, as you guys know. But that's still a pretty small for all of that material. So all of the structures are there in miniature form. are all the neurons communicating with each other? No. And are all the parts, we're going to talk about the anatomy of the neuron, are all the parts of the neuron developed? No. So that's what is to come, right? And actually, prenatally, when your mother was pregnant and as your brain was developing, and it happens, so early in prenatal development. That's why it's kind of scary. You may not, a female may not even know she's pregnant, and the brain stem, it's called the neural tube. The neural tube is exposed, and about 6 weeks pregnant, that neural tube closes and then further develops into the brain and the spinal cord. So it's vulnerable to toxins during that time. So if mom doesn't know she's pregnant and she's drinking and smoking and doing drugs, breathing in paint and toxic fumes in her work environment, it could be really, really bad, right? So as soon as you find out that you're expecting, you want to eliminate all those toxins. Hopefully you've been taking a prenatal vitamin or folic acid to protect the neural tube. And if you drank one time in that early part of pregnancy, you're probably okay. But you just want to stop whatever that hazardous behavior is, and so that you can protect this developing baby's brain. At a certain point in prenatal development, you actually have about 300 billion neurons, but that's too many. And so then the unneeded ones die off and they prune, and then when the baby's born, they have all their neurons. But their nervous system is very immature. And that's why babies sort of twitch and flutter when they're sleeping. They can't control their limbs. They're only born with the very basic reflexes. So it absolutely just takes maturation, which is the growth time going by for that brain to develop. And then, of course, nutrition in a stimulating environment. They have to be cared for by others. Right? Okay, so the basics of the neuron then. First of all, let's talk about the different types. So I said we've got about 2/3 of them are densely packed in your cerebral cortex, right there in your head, and the rest go down your spinal cord and then out through your entire body. So we have sensory neurons, and as you can imagine, the word sensory is bringing information in through your senses up to your brain for you to process them. So we have lots of different type of sensory neurons. You have touch receptors on your skin. We have specialized sensory receptors in our eyes, at the back of our retina, cones and rods, right? Those are the specialized receptors. In your ears, in the cochlea, you have this little coiled tube, and inside there are these little hairs called cilia. Those are sensory receptors then that basically sound comes in through sound waves, which is air moving, right? Comes into your ear, gets amplified by the eardrum, pushes against those little hairs, and right to your auditory nerve, and then your brain understands it. So all of these different things are sensory neurons, information coming in through your senses. And then your motor neurons that's your actions going out, right? So your brain telling a certain part of your body to move. Some of this is voluntary, some of it's involuntary. A lot of it we can, with time, we just kind of do it automatically, but we can also stop ourselves from doing it. And then some of them actually become reflexes, where we're integrating sensory and motor neurons. And of course, interneurons then are the third type, which are the go-betweens. So we mostly have interneurons. They're the go-betweens between motor and sensory neurons. So for an example, thinking about a reflex response, if you touch something that's really hot, okay, so you touch the hot stove, and you're going to get an electrical impulse, right? Sensory neuron from your finger is going to run up through your nerves, through your arm, and actually to your spine, and you're going to fire neurons actually out of your spinal cord to pull back. And then you process it. So it's actually, your body actually moves and reacts faster than your brain can. So that's where your spinal cord is part of your central nervous system. It would take too long. By the time the signal got all the way up between the interneurons, all the way up to that part of your brain, your finger now would be very, very badly burned, even worse than it is in that second that it took you to pull it away when it goes to your spinal cord. Other things, will travel all the way up to your brain. You process them, and then react back down with moving your body. Okay. And like we said, we have about 100 billion. That's a rough estimate, clearly. But this makes lots and lots of connections. So if you think about, you're born with all of these neurons, but a newborn baby can't do much. They have their basic reflexes. Sometimes we forget about the really obvious reflexes like breathing and swallowing and sneezing, coughing, certainly heart beating, and those are all basic reflexes. Sucking, but then they have some other reflexes that are sort of probably precursors to movement, like grasping. If you ever put your finger in a newborn baby's, boy, they grasp hard. Well, that's probably, that's the pre-wiring, getting ready to actually be able to use and manipulate their hands with purpose, as opposed to just being a reflex. So as that developing baby is gaining new experiences and then just having the natural maturation of her brain growing and getting nutrients from the breast milk or the formula, she's creating new connections. You can have 1,000 connections to 50,000 connections per neuron. We're talking trillions of combinations. And it's not like you just have neurons that are neighbors that are communicating with each other. You could have synapses crossing, from, the left hemisphere basically controls the right side of the body, the right hemisphere controls the left side of the body, and receives information that way. So you've got information running all the way down your spinal cord, down your leg, to your toe. There's this massive network of communication. So our basic structure of the neuron then, see if I'll draw a picture here. The cell body is very irregular in shape, right? It's not like skin cells that are kind of a rectangle shape, or, when you look at them under a microscope, you can identify them. Liver cells, lung cells, they all have a particular shape, or like red blood cells, they look like donuts, right? But a neuron has a very irregular, sort of rough edge. It almost looks like a globby bit of snot, and it's kind of gray or whitish in color. But it does have a cell membrane around it that's holding all the nutrients in there, holding all the information in there. And it does have a nucleus in the center there somewhere, a dark spot. And what's housed in the nucleus? The DNA. Good. Right? So you've got 46 chromosomes, 23 from your mother, 23 from your father. Only the genes that activate the neuron cell are, sort of awake within the center of that nucleus. But you have a copy, the same exact copy of the chromosomes in the nucleus of every cell in your body. Right? Just different genes are activated in those particular cells depending on what the purpose of the cell is. So you've got other, different things going on inside the cell body that nourish it. But for the purposes of this class, you don't need to know maybe all of that terminology. You'll get to that if you take anatomy and physiology or a biology class. All right, so then we have all these bushy fibers that branch off everywhere, extending out from the cell body. And what are those called? Dendrites. Dendrite is Greek for tree or branch. So they are these bushy fibers. And at birth, there aren't tons of dendrites. With new experiences, we grow dendrites. the anatomy of your brain changes with experience. So if I ever say, hey, are you guys growing dendrites? That's my way of asking, are you learning? Are you actually, having something new happen in your brain? Or is it just going to go in and out? Because if it just goes in and out, you're having some chemical release because you're listening and maybe processing, but at kind of this real surface level, and then you immediately forget it, or you forget it right after the test, and you didn't really change your brain. So to really change your brain, we have to have this structural growth of dendrites. Okay? So dendrites are the bushy ends, and then you have this long cable called the axon. Now, some neurons will have 2 or 3 axons. For the purposes of this class, I'll say it again, let's just try to remember 1 axon. Just makes it easy. Okay? 1 axon, lots of dendrites, thousands of dendrites, 1 axon. And at the end of the axon is the axon terminals. They do kind of branch off into these little terminal buttons. And your axon has a coating, a fatty coating there. And what's that called? Myelin sheath. Okay? So the myelin sheath is a protective coating, and it's actually built up by the glial cells. So if you have 100 billion neurons, you have about 9 times as many glial cells in your brain. And what the glia are kind of like helpers and workers. They nourish the neurons. They build up this myelin. They feed the cell, right? So they're helpers and workers. If it weren't for the glial cells, your neurons wouldn't really be able to communicate. So what else does the myelin sheath do besides protect it? Speeds transmission, right? So it's this fatty substance, so think of it as being like slick. So it makes the neural impulse go faster. So with more and more experience, say you're learning how to kick a ball, and your little developing 3-year-old brain is watching that ball roll at you, and of course anybody who's ever watched kids that age, they're very coordinated, and they kick too late, and they miss. Well, so then they keep practicing and practicing, and it's just a matter of time. It's maturation of the brain where they'll be able to coordinate the movement with controlling their foot. And then if they practice it over and over, now maybe the child is 7, 8 years old and they're really into soccer, if we're talking about kicking a ball, and they practice it over and over and over again, now they've got that pathway from the visual pathway to the large motor movement and the timing, and they're coordinating that, and they're doing it over and over and over again. You've got a neural circuit then that's been practiced a lot. So we've got lots of myelin that's been built up in that circuit. So the child is better at it and faster at it. So even if you haven't done something for years, if you laid down some pretty strong pathways in the early part of development, then it's going to be easier to get it back. Even professional athletes have to keep practicing, but if you learned it really solidly the first time, even if you've been away from the activity for 10 years, or say, let's go to like foreign language. Say you haven't studied the foreign language for 10 years, but you did learn it pretty decently back in the day, and you laid down the neural pathways, and you practiced and you recited, and you knew how to conjugate a verb and do all that stuff, and now it's 10 years later. Yes, you're going to have to practice again, but it's probably going to be easier because you laid down the pathway, and you just need to kind of prod it and get it going again. Okay. So those are the major important parts of the neuron. Here's a nice picture there, a little bit better than my drawing, right? So all of these different things that nourish the cell, the mitochondria. I don't even think Myers has that as vocab words in your book. So we're pretty much looking at cell body dendrites, axon terminals in the myelin sheath. The axon hillock, that's the gate. That's the gatekeeper. Whether this thing is actually going to create a neural impulse or not is up to the axon hillock. And I'll get to that when we talk about neural communication. Here's a nice little drawing of the dendrite. You can see in relation to the size of the cell body how many dendrites a single neuron can have. And there, look at how in proportion how small the axon is. So you can imagine if you have this, neurons are microscopic. You can't see them with the naked eye. If we were to look at a brain, it would just be gray and white. If we were to look at nerves, which are bundles of neurons, again, they would be sort of silvery or white in color. Don't touch them if you ever have an injury and expose a nerve. Yeah, ouch, right? Luckily, the surface of your brain doesn't have nerve ending receptors, so you can actually be woken up during a brain surgery and it wouldn't hurt. Of course, they'd have your skull all anesthetized, right? But it is possible to do. So you can't see a neuron without looking. You need a microscope to see the neuron. But you can imagine how this tiny structure then, with all of those dendrite branches, how that can capture so much more surface area in making really complex things happen. Right? Think of this network that's going on. And that flurry of development is biggest in the first 3 years of life, and then things kind of stabilize. And that's why the baby's head is so out of proportion. Their head is huge compared to their body, right? They can kind of hold up their head and it flops over. And why do you think they call it a toddler? The toddler has short limbs and a big head, and they toddle around, and their head will whoop, 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 right? Because their brain is still too big. It's actually 50%. At age 2, it's 50% its full weight. So this is very, and a 2-year-old is like maybe 3 feet tall, probably 2.5 to 3 feet tall. So it's a pretty big structure that they're toting around on their body. And then things kind of stabilize a little bit through childhood. You'll notice the child's body sort of catching up with their head in middle to late childhood. And then adolescence happens, and their body and head will be getting towards full growth. But then we have another flurry of brain development with this dendrite growth. A 5-fold increase in dendrite growth at adolescence, and mostly in the prefrontal cortex. So this is where all of a sudden the child can do complex problem solving, think logically. They can do algebra, that hypothetical deductive reasoning. But it's almost like it's so much growth so fast that they can't organize it. And so they're really impulsive. Oh, you never heard of that? Teenagers being impulsive? So they're like really smart and creative and passionate and have a lot of capacity, but then it's not organized. So then they might make rash decisions. And they live in the moment. A lot of you guys are teenagers still, so I think maybe you know what I'm talking about. Okay, so the axon, that long cable, is covered in the myelin sheath. It protects it, insulates it, and then also speeds transmission. Here's all these little axon terminals that branch off at the end. So certainly not as many as dendrites, but it's not just like one little structure at the end. Okay, so I use a lot of interchangeable terms, and I've already said, electrical communication, synapse. I might say, your neurons are talking, something's happening. All of those different sort of terms. What we're talking about is an action potential. And action potential takes place when we have a change in electrical charge. Okay? Now, this is not like electricity in your brain. Okay, electricity moves much, much faster than what we're talking about with our electrical activity in our brain. And it also isn't quite like plugging in a cord and electrons hopping. Has anybody taken an electrical engineering class lately, or you've learned it in physics, right? So it's not, that's kind of an okay analogy when we're talking about electrical activity in the brain, but it's not exactly the same thing, because it's not like the chemicals are literally passing through your neurons. They are just exciting the neurons to open up channels. So your neuron has all these different channels. It's going to allow ions to pass in and out of it. So when a neuron is at rest, when it's not doing anything, it's mostly negatively charged. Now you have negative and positive ions inside and outside the neuron, and inside and outside that includes the axon, of course. And you've got all this cerebral spinal fluid that your brain is sort of suspended in, right? And so that's also out here. So when it's at rest, it's mostly negatively charged on the interior of the neuron and positively charged on the exterior. And as you learn more about anatomy and physiology and in your bio classes, you'll learn about the exchange of potassium and sodium ions. But I know that Dr. Myers in this textbook really keeps it very simple. Let's just break it down to negative and positive ions, because they don't even ask you those types of test questions. When you take biopsych, if you go to the University of Washington or Western or WSU, then you'll get on a sort of a higher level, right? But I think for this class, let's just keep it to negative and positive ions. So it's mostly negatively charged. So when it's at rest, we say it's polarized. And they measure it in milliseconds. So compared to a computer's in nanoseconds, right? Like really fast. So our brains, while very complex, not as fast as the computer, right? We react a lot slower. Something happens that you need to react. Sometimes our reaction is too slow, and then you know you accidentally get hit on your bike, right? Because your reaction is too slow because the car is faster than you, right? You can't react. So when it's at rest, when nothing's happening, it's about negative 70 millivolts is how they measure it, right? So something's going to come in and excite this neuron. It's always coming in through the dendrites, right? The dendrites are always the receivers. So, of course, your dendrites can be in any direction, branching off of the cell body, but the information is coming in, and it can be in chemical messengers or electrical messengers. I'm mostly going to talk about chemical messengers because that's what we're interested in psychology. It's what changes our consciousness, and it has to do with our reward system as well. So the information comes in, and it causes these channels to open up, and the ions rush in, positive ions rush in, and negative ions rush out. And if it's enough to excite it, then the axon hillock will open up, and then you'll have an ion exchange take place running down the axon. So this is where it's not like electricity. Electricity, you have electrons hopping, like down your cord, but what's happening here is your ions are rushing in and out down the axon. Right? And then it's going to cause something to happen. When it reaches, it can get up to plus 40 millivolts then in the change in charge. So an action potential is a change in electrical charge from mostly negative to mostly positive, and we call this process depolarization. So some interchanging vocabulary. Okay, so at rest it's polarized, and action potential it's depolarized. Okay. So then you ask, well, what happens then when you have that change in electrical charge? Well, let's draw a really big axon terminal. Okay, so inside your axon terminal, you have these little fluid-filled sacs. And what's housed inside those sacs? Really, neurotransmitters, good. Really special information. So neurotransmitters are in there. And that's where they hang out when nothing is happening. You have an action potential take place, and you've had an ion exchange from mostly negative to mostly positive. It's going to cause these synaptic vesicles to open up and release the neurotransmitters out into your brain, out into that fluid-filled space. But then, of course, it's going to want to try to latch onto something, which would be a neighboring neuron, right? So your neurons don't touch. Now, even if we looked at some neurons under a microscope, it would be very, very hard to see that they might look like they're touching, but they're not. They can be a millimeter apart. They could be quite a ways apart. If the neurotransmitters can float through your cerebral spinal fluid there and dock onto the receptor sites, then, we'll have a synapse take place. Okay? So this little gap then is called the synaptic gap, right? Or cleft. Sometimes you'll read that in a book or in an article, the synaptic cleft or synaptic gap. And then this process of crossing the synaptic gap, neurotransmitters crossing the gap and then docking onto a receptor site, is called the synapse. Okay, so I hope that makes sense. The gap is the space. The process of the neurotransmitters crossing is the synapse. So sometimes when we say action potentials are taking place, we may also sort of interchangeably say synapses are taking place. And that's the real important term because that means something is happening. You actually have neurotransmitter release docking on to a neighboring neuron. Okay, so if this is an axon terminal here that's sending, what part of a neuron do you think is this one down here that's going to be receiving the chemical messengers? The dendrite, right. So axon always sends, or we can say axon away, AA, axon away. And then this is going to be the surface of a dendrite, receiving. And they lock right on there. And then maybe they're going to cause ion channels to open up in this neuron. And that's the chain just keeps going throughout your brain and nervous system. So in my little drawing here, which one is the presynaptic neuron? This top one, right? And which one's the postsynaptic then? This one down here. All right, so dendrites are always receiving. Axons are always sending. It cannot go the opposite direction. So then what happens to these neurotransmitters after they've docked on and maybe they've created another action potential in this neuron? What happens to them after they've docked on there? Where do they go then? I know some of you have studied this before, I could tell. They actually go back into the sending neuron. So they dock on there onto the receptor sites of the dendrite, if they're excitable, right, if they're going to cause an action potential to take place, then we're going to have some more ion opening up, ion exchange in this neuron. And then they're going to go back from where they started. They don't actually travel through the neuron at all. They are always housed in those synaptic vesicles at the tips of the axon terminals. Information, they say, okay, open up the ion channels. Or they could be inhibitory. They could put on the brakes and say, okay, don't do anything. But then they're going to go back up. And this process is called reuptake. Okay? And there's a refractory period. There's a resting time before anything can happen again before it can get excited again. It needs to rest. And that could be a fraction of a second, or it could be several seconds. Did you have a question?
Yeah, when you said there's an ion exchange when it goes to the other dendrite, does that go to the next cell, or does there like a impulse that goes to the next cell, or does nothing happen in the dendrite when it goes into?
Is there an impulse?
Is there something that happens in the dendrite that goes down the dendrite, or does nothing happen with that? Like when it exchanges the neurotransmitter, does anything happen at the other end when it's exchanging?
At the axon terminal end?
Dendrite.
At the dendrite end. So the dendrite end is just receiving the messages and then opening up those channels to the cell body. If it's enough information, like you could have, potentially there's lots of different axon terminals that are sending out neurotransmitters that are exciting a single neuron, right? It's not necessarily, or this could be exciting several neurons, right? So you may not have enough for anything to happen at all. In that case, nothing happens here. This neuron doesn't get enough of an ion exchange for anything to happen, and those chemical messengers, those neurotransmitters, are just going to reuptake back into the sending neuron, and nothing happens. If it's enough, that's up to the axon hillock. The axon hillock gets enough of excitement, change in charge to positive charge. It opens the gate and then runs down the axon. So I think when I took this class, back at the University of Washington, I think I was a sophomore when I took it. I didn't understand that either. I was wanting to visualize in my mind the neurotransmitters like gobbling down, like they were moving. And that's not what happens. They really just absolutely stay in this. They are housed in the synaptic vesicles. They are released, excite, or inhibit, back up. So they just kind of go back and forth. Now, if we have a drug like cocaine, okay, so you take cocaine. What cocaine does is it stimulates a lot of different areas of the brain and excites a lot of neurons to release these neurotransmitters. So it will excite your brain to open up your synaptic vesicles and release all sorts of serotonin and dopamine. And so then you have all this dopamine, too much dopamine, that has no place to lock on to neighboring dendrites, so it'll just kind of keep pinging around in your brain looking for other receptor sites, and it kind of floods your frontal lobe, which then is going to cloud your judgment. If you do enough cocaine, don't do cocaine, okay, but here's my example. If you keep doing cocaine, then you actually could have a hallucination, and you actually would exhibit sort of psychotic-looking behavior. You have hallucinations, and you think things are there that aren't really there because you've got so much dopamine and serotonin pinging around in your brain, sort of with no place to go, and you get euphoria is one name for it, but then it could be, over-the-top paranoia then, right?
Does it damage your receptor?
Yes, good question. So over time, any drug that you're introducing into your body is going to damage your receptor sites. So alcohol also indirectly stimulates the release of dopamine, and when we get to the major neurotransmitters, dopamine is definitely one of them. The dopamine is like our reward system. Anything that you perceive as being pleasurable means that you are releasing dopamine from the center of your brain and then flooding out to your frontal cortex. So if you like strawberries and you're eating strawberries, you're releasing dopamine. Okay? If you like alcohol and you're drinking alcohol, you're indirectly chemically releasing dopamine. But then if you become addicted to it, now your naturally occurring dopamine goes, oh, she doesn't need me anymore, and kind of gets silenced, doesn't really react, and you've had all this excess dopamine, and you damage your receptor sites, and those are permanently damaged and altered. That's why they say once an alcoholic, always an alcoholic.
So they don't grow back?
No. So then you go on antidepressants. Yeah. Yeah, a lot of people, after they've been through drug rehab, are depressed because maybe they've regained some of their dopamine system and their serotonin system. They've regained that, but there's no place for it to dock on, and if you're an adult, your brain is, more fixed in its pathways, so then you go on antidepressants to try to recycle what little amount of serotonin that you have in your system that you're able to kind of try to recapture so that you don't stay in a deep depression. Yeah, so definitely introducing substances into your system can have alterations to, how synapses work. Yes, question. Is there, yes, that would be called schizophrenia. It's actually it's a hypothesis. The dopamine hypothesis of schizophrenia is that you have an excess of dopamine, and this is probably inherited. Schizophrenia tends to run in families, and they study identical twins, and if one twin has it, the other twin has 50% likelihood of also developing schizophrenia. So it's probably something chemically about that you've inherited, and you have too much dopamine, and that's why someone who has a drug psychosis, too much dopamine pinging around in the brain, if you don't know what's going on with the person, they look like they're schizophrenic, or they might look like they're having a manic episode, like a bipolar person would. It's hypervigilant, it's agitated, it's excited, or violent. It kind of depends on the personality of the individual. There is a gene also where some people naturally have higher levels of serotonin, and then that would be good. You want to inherit that because then you're kind of always calm and relaxed. We relate that to temperament. So those easygoing babies that you're able to get them to stop crying pretty quickly. You pick them up, you hold them, you soothe them, you lay them back down, and we label them easy babies. They might have that gene that's producing a higher level of serotonin, and they kind of loosely named it the happy gene. But that would be a good one to have, and it's more about serotonin and just being calm and relaxed, not being hypervigilant. The hypervigilance, that's dopamine, the overexcitable. And you had a question.
Yeah, when the neurotransmitters cross the gap, what exactly happens? It depends on whether it excites or doesn't excite.
So it depends on, so she's asking when it crosses the gap, what makes it excite or not excite? It depends on what type of neurotransmitter it is and what the function of those neurons are. So that's a really great question. It kind of leads us into talking more about neurotransmitters. So we have this wonderful natural painkiller that our brain produces called, anyone know it? It's one we haven't said yet. Endorphins, great. Okay, so endorphins are natural painkillers. Endorphin means opiate-like. So you can, this is where this understanding and learning the basics of neural communication and neurotransmitters is really important because we'll keep coming back to it when we talk about drugs and consciousness and psych disorders. So someone who does heroin, which is a synthetic man-made version of morphine, and morphine you get from the opium poppy. It's opiate-like, so it's a painkiller. So we give morphine to patients in the hospital when they're in severe pain to block the perception of pain. So it would be a message that would then block this nerve cell from firing. Okay, so we have natural endorphins in our brain. So you stub your toe. It hurts. It's kind of funny. You stub your toe, you don't feel it. A couple seconds later, you feel it because that's how long it takes for the message to travel from your toe up to your brain. And then you feel it, and then it's throbbing, and then you wait. Okay, maybe it starts to subside if you didn't get hurt that badly, right? It starts to subside. What is that that's doing that? That's the natural release of endorphins in your brain that's then blocking that firing down to your toe. You look down at your toe, it's still red and throbbing and bleeding. It's still hurt. But your perception of it being hurt is blocked because of the neurotransmitter endorphins that are telling this motor neuron to stop firing, right? So it's really kind of interesting. It's just a perception of pain.
So would that mean that junkies that their endorphins don't work anymore?
Yeah, so possibly, and then if you stop doing drugs that are, like if you stop doing heroin, right, you'll get these terrible withdrawals, and your body hurts and shakes and everything because your brain hasn't been producing endorphins naturally for quite some time. That's where your brain becomes addicted to the substance. One really great way that we can get endorphins to release through our system is to do what activity? Exercise, right? Exercise is a great way to produce endorphins. And it opens up those blood vessels throughout your whole system. And yeah, it's reducing pain, but actually then, have you ever experienced that moment in your workout where you're hurting and then you kind of go over, just it's like cresting the hill, like literally you could be cresting the hill, and all of a sudden you get a burst of energy, we call it the second wind, and your whole body just seems to be so efficient, and your oxygen capacity goes up. And if we were to literally measure what's happening in your brain and in your body, we would see that that was where the endorphins have been released and have become real efficient throughout your whole body. Now, it takes a while to get there, right? You have to get in shape before that to happen. But then once you get into shape, you don't want to stop your exercise regimen because it'll hurt again building up to that level of fitness. But we know that release of endorphins in your system then is going to also encourage good production, steady production of dopamine throughout the day. That's why doctors almost always prescribe exercise for people who are suffering from depression. You got to move your body. Move your body, move your body. And it's not just to so that you're not at risk for harmful behaviors. It literally can boost your brain activity and really make you feel better. We had one more question in the back.
I was just wondering why so
Oh, wow, yeah. So it's a very powerful drug. Morphine is derived from the opium poppy flower. It's a substance that they discovered, and somebody at some point probably ate it or smoked it, and were like, wow, that feels good. And so then, but it's used in medicine. It's a very powerful drug. And because it's so powerful, it alters that chemical system in your brain. So that's why usually doctors they'll want to only prescribe it for a certain amount of days. Or since we don't know what our own brain chemistry is, you don't really know. Everybody's brain chemistry is unique, and you may not necessarily know if you're prone to addiction or not. Maybe look in your family, like if you've had anybody, and especially only one generation away, but even 2 generations away, if you've had people who are alcoholics, or that means that you might be more susceptible to addiction. And the more powerful the drug is, then the more susceptible. So we see methamphetamines probably the most addictive. That can respond to a person's brain after one use, create the cravings, those cravings, feelings. Like something like caffeine. Okay, I'm addicted to caffeine, and I get this sort of craving for the taste of coffee, or you're like, I want coffee. And then you go into the coffee shop and you smell the coffee roasting and you go, okay, and you're already starting to feel a little bit more awake and alert and steady and calm, and you go, wow, I'm addicted to caffeine. It would take me about 3 to 4 days to get off caffeine. I would get headaches, would maybe even feel a little bit sick or nauseous, shaky, but it's not nearly as strong a drug chemically in altering your brain as morphine. Yeah, and then your individual, unique biochemistry. Yeah. One more question, then I'll keep moving on.
Stemming off of the caffeine addiction, would you consider decaf coffee like a placebo sort of thing to be able to trick your brain to get off the caffeine?
Yeah, decaf's a good way to taper off. There are minute traces of caffeine in decaf coffee, and you have to be careful because sometimes the servers mess up and accidentally serve you caffeine. But a good way to, if you really want to get off the caffeine, a good way to taper off is go half and half regular, half decaf, and then do that for a week, and then down to decaf, and that will eliminate some of the withdrawal symptoms. And then you still get some people just like the taste, and then you go, oh, okay, I'm still getting my taste of coffee or the warmth. Some people really just like having something warm to drink in the morning. Caffeine's fairly safe. It's the most common psychoactive substance in the world. Almost every culture drinks it, either in coffee or tea. So it's pretty safe. Don't go crazy. Like less than 5 cups a day, and it seems to be pretty safe. If you're pregnant, there are some different guidelines, maybe 1 to 2 cups a day if you're pregnant. Okay, let's keep going here. All right, here's a nice picture looking at the action potential. So you can see you've got your axon terminal. The synaptic vesicles are those little bubbles there in the middle that house the neurotransmitters. When you have the ion exchange, that is happening through the surface of the axon, that's going to cause those vesicles to open up and release the neurotransmitters in the space, the synaptic gap. And the neurotransmitters, the chemicals there, are particular shapes that then fit onto the receptor sites of the receiving dendrite. Okay? And there is no bolt of lightning, electricity. I'm almost always depict that in graphics and when you watch videos. Your brain is not lighting up like a light bulb, right, and like snapping and popping, right? What's interesting, though, when they do imaging studies, when they do the functional MRI, the technician or the doctor or scientist can isolate what part of the brain they want to look at, and then on their computer they will color code it. And when they see electrical activity take place, they are coding it in some kind of, they'll give it a color. They'll say, okay, all the dopamine centers we're labeling green or whatever, and then they look at that on their computer screen. So yeah, they are sensing, and the PET scan is also picking up that electrical activity. And they will say the area of the brain is lighting up, but it's not lighting up like fire, like electricity. Okay. All right, let's do a little review. Which is the sending part of the neuron? Three, 2, 1, closing. Okay, great. Axon away, that helps, right? I always like to come up with little ways to remember things, okay? Axon is always sending. All right, so then what's the receiving part of the neuron? 3, 2, 1. Great. Almost everybody's got it there. Okay. Dendrites are always receiving. Okay. What is an action potential? Three, 2, 1. Okay, pretty good there. All right. So an action potential, we've got depolarization taking place. Your charge goes from mostly negative on the interior to positive on the interior because of that ion exchange. Right? Great. And so your axons, the axon terminals are branching out and ending near neighboring dendrites. It's really a complex network throughout your entire cerebral cortex and down your spinal cord and then throughout your peripheral nervous system. The axon terminals are the tips, these little tips, these little branches, and they have the synaptic vesicles inside there with your neurotransmitters housed in there. The gap or the cleft separates the axon terminals from the dendrites. And when we have that exchange take place, that gap, then that's called the synapse. Okay. So in the action potential, I don't know if I've mentioned this, it's an all or nothing process. So if you have the axon hillock, the gatekeeper, open up, so you've got enough of an ion exchange in the cell body, and the axon hillock has opened up, and now you're going to have ion exchange run down the axon, it's going to go. As soon as that hillock opens up, the action potential takes place. It can't go halfway down the axon and stop. It can't do just a little bit and stop. It's all or nothing. If the gate opens, an action potential takes place. Okay? It's non-decremental. That means that it doesn't speed up or slow down. It's the same speed the entire way, right? The exchange of ions going down the axon. So you might ask, well, how fast does this happen? Well, it kind of depends on where it's located, what kind of neuron we're talking about here. But as I mentioned, it's measured in milliseconds, right? So it's not super fast. There can be some that move at about 180 to 200 miles per hour. That's pretty fast. But there can be some that are as slow as 2 miles per hour. That's a pretty slow. You're like, wow, that takes it. That's probably not your thought process. Let's hope your thoughts don't move that slow if you're trying to solve a problem, right? But I mean, even just language, once you come to really learn a language, you're in elementary school, you're very proficient at carrying on conversations and speaking. You don't have to really word search too much. You can, once you've learned the rules of grammar, which just happens naturally, by the way, speaking. Now, reading and writing in the rules of grammar, that's a process, right, that you learn in school. But speaking, whatever your native language is, speaking and using those rules of grammar, it's pretty amazing how the child will just do it and apply the rule, and then they over-apply it. So they see a mouse, and they see another mouse, and they look at the book, and they go, look at the two mouses. Because they haven't learned the irregular rule, their brain so they just naturally apply these rules. They're not directly being taught. It's pretty fascinating how children pick up language. So as soon as you develop that, maybe talking and thinking, maybe those are the things that are happening pretty quickly. Now, mathematical computations, I don't know about you guys, but for me, that takes a little bit longer as I'm processing that. But maybe my reflexive response of touching the hot stove is one of the fast ones, okay? But it's non-decremental. It doesn't speed up or slow down. There's a refractory period. That's the resting period between when this neuron can fire again, when another synapse can take place. So you've got reuptake back into the sending neuron, and then it has to rest before it can be released again. It could be, milliseconds, or it could be 30 seconds. It depends on the location and purpose of the neuron. The threshold is whatever point that's reached in the interior of the cell body. Once you've had enough ion exchange and it's gone to a positive 40 charge and it opens up the gate, you could still have more neurotransmitters latching on to that neuron and have more ion exchange taking place, but it kind of doesn't matter. It's like you don't need that much. It's already opened up. It's already fired. It's reached its threshold. So once it's reached the threshold, it goes. And here we have the actual neurotransmitters, which are chemicals, right? And they have particular molecular shapes. If you take a chemistry class, you'll learn all about that. You won't need to know that in this class. You also won't need to know the abbreviations in this class. They always spell it out in the test questions. But this is norepinephrine and this is acetylcholine. But they always spell it out. So that's just my picture there. So they have particular shapes. And it's kind of like a key in a lock. And they fit right on there. Okay. And either you've got it binding and ions are entering, and you've got something going on. It's going to create either an excitatory message. So if it binds to the receptor site and it allows those ion exchange to take place, opens up the ion channels, then it could be an excitatory message, or it could put on the brakes and be inhibitory. Right? And then, of course, we've already talked that you can take substances into your body that mimic these naturally occurring neurotransmitters to cause things to happen in your brain. Okay, so our important neurotransmitters then. We've already mentioned several of these. You'll hear me talk about these over and over and over throughout the quarter. I will occasionally talk about a few additional ones, but these are the real important ones in psychology. GABA, gamma-aminobutyric acid, that might be one that's new to you. Acetylcholine, dopamine, endorphins, serotonin, and norepinephrine. So we've mentioned a few of these already, but let's go through these one at a time. So GABA, gamma-aminobutyric acid. This does something in the brain that is pretty important. And it's not about sleeping like this guy on the toilet, because clearly this person probably in his right mind wouldn't have chosen to sleep here. What state do you think got him here? He's drunk. He passed out. This is why you don't put pictures like this on Facebook, because then they get on the internet and then people like me pull them off. I have no idea who this is. Okay, so, or of your friends, right? And then your friends like hate you forever. Okay, so now alcohol doesn't have GABA in it, but alcohol indirectly stimulates the release of GABA. And what GABA does is it slows down your brain activity. What kind of drug is alcohol? It's a depressant. Good. It's a depressant. So, when you have a drink of alcohol, you've had, one glass of wine or one beer, maybe you're on your second drink, you actually get sort of talkative. right? Maybe a little giddy, maybe a little uninhibited, right? You might do or say things that you wouldn't ordinarily do, but it's not because your brain is being stimulated and awakening those areas. It's actually because it's hitting your frontal lobe that's slowing it down. The GABA is being released. It's slowing down your prefrontal cortex here that's right behind your forehead. And that part of your brain is executive functioning, decision making, logic, reasoning, planning. So you stop going, oh, wonder, like, if I say this, it might hurt someone's feelings, or your judgment gets slowed down, right? So that may come across as being that you're, looser and giddy and more talkative, but then eventually, if you keep drinking alcohol, then you're going to have more GABA flooding, and of course, we're also indirectly releasing dopamine, which is that elated feeling. So then you start hitting further back on your cerebral cortex, and now you're hitting your motor cortex. So now your motor movements are a little bit slowed down. And then you've got your language centers that are kind of here on the left side of your frontal and parietal lobes. And so now literally you're talking and your words might be slurring. Now you're getting pretty drunk, right? If you're hitting that point. And then eventually it's going to flood to the back of your brain. Back here you've got your cerebellum, which is that little bump on the back of your skull. You have a whole other structure that sits below your cerebral cortex. And that has to do with, also with decision making, but balance and coordination. Okay? At this point, person might not pass that test. You might not pass the test further, right, when you're to walk the line or to touch your nose with your eyes closed. But you've got so much GABA flooding throughout your brain now, your reaction time is really impaired. So you shouldn't drive or operate machinery, right? And then, of course, if you keep drinking alcohol, where's the next place it's going to go? Back deep inside, below your cerebellum. We haven't studied it yet, so that's okay. It's going to hit your medulla, your brain stem. So you could die of alcohol blood poisoning, right? So that's your heart, your breathing and heart rate. Some people, if they get to the point where they've passed out, they actually could die from asphyxiation on their own vomit if they vomit while they're asleep. Yeah.
What happens when you take GABA from the store or something? Does that create that much GABA? Or?
Somebody was talking about that last quarter, and I came across an article, and I don't have a precise answer for you right now, but I want to say that it's somewhere along the lines of that it's not really doing anything. It's not enough to help you sleep. You would say, why would you want GABA? Well, there are times that you want your brain activity to slow down, right, so that you can relax. You want to stop thinking about everything that happened to you that day so that you can gear down and fall asleep at night, right? So some people maybe would say, oh, we'll take a GABA supplement. I don't know that it clinically, like, what's the science? I always say, what's the science behind it? Have they tested it? Have they done double-blind studies? Have they tested it on animals to see if it actually does anything? Have they watched it on a neurochemical level to see? So I don't know, but I do kind of remember hearing something about that. Always be a little bit cautious of all those supplements. They're so expensive too, so then you kind of go, what's in this? Yes, question? Yeah, someone with epilepsy, the drugs that are prescribed for that to control seizures there's so many different types of anti-seizure medications out there, and some of them they don't know why they work or exactly how they work, neurochemically, what exactly is going on. It's essentially keeping the neurons from firing at will. Because that's what a seizure is. The neurons just fire on their own for some unknown reason, And sometimes they are activated by alcohol or lack of sleep, or somehow your brain is a little bit off balance, and then a seizure can be triggered. Bright lights or strobe lights can do it for some people, but other times it's unknown. It just happens. And so there's lots of different types of drugs. I'm not sure if GABA is indirectly triggered by some of those different anticonvulsants. I'm not sure. Okay, I want to keep going. Okay, acetylcholine. Acetylcholine, we see this in a part of our brain called the hippocampus that's really being produced there. And what is this little shell game a test of? It's a memory game. So acetylcholine is essential for forming new memories. And we see that being produced in the hippocampus. And what disease is associated with memory loss? Alzheimer's disease. So we actually now have the technology to look at a living, working brain and see these plaques and tangles building up that are indicative of Alzheimer's. The PET scan actually is the technology that's used to be able to see that at that level. And they see these plaques and tangles building up in the hippocampus first. And so that's where, and we see depletion of acetylcholine in Alzheimer's patients. Now they've tried to give supplemental acetylcholine, really hopeful, thought, oh, this is great. We now finally understand that acetylcholine is essential for producing memories and making new memories. And so maybe we can give acetylcholine supplements or drops to people. And they had a clinical trial, and it worked well in the rats, so then they gave it to the humans. And this was about maybe 10 or 15 years ago. And then some people died of brain swelling. So it didn't work. And now they're trying something with doing like nasal drops, and they're still testing on rats, mostly rats and mice. There's so much research out there on Alzheimer's to try to figure out how can we either get rid of the plaques that are building up in the brain, or stop it from building up, eliminate it from happening. And but acetylcholine then is what is released as we're learning. So we're learning, we're creating a memory. That's facilitating that process. Very important. Okay, so dopamine, we already talked about dopamine being your reward center. Anything that you are doing or experiencing and you're getting pleasure out of that, we know dopamine is being released. Okay? And it's being released from the caudate nucleus, which is the deep center of your brain, and then it kind of moves forward. The nucleus accumbens, and it moves forward through your frontal lobe. But this guy is doing a balancing exercise, and he's not smiling. So let's say that it's not that he's enjoying this. So what else do you think dopamine is really important for? Not quite. It's pretty basic. Just absolutely motor control, motor movements. So if you had low levels of dopamine depletion, you would have a really hard time doing this exercise that takes a lot of balance and coordination through all your large motor muscles there in your trunk and through your legs and his shoulders. He's holding his shoulders upright. That takes a lot of control. So there is a disease that's associated with depletion of dopamine. Can you take a guess? Parkinson's. It's a neurological disorder, and it's progressive. And if you've ever known anybody with Parkinson's, they will be shaky, right? Their motor movements, their muscular control. Researchers have had greater success in supplementing dopamine for the purposes of motor movement with a drug called L-Dopa. And you guys know Michael J. Fox, who was diagnosed with Parkinson's at a fairly young age. I don't even know if he was 40 when he was diagnosed, very young. And he has been on L-DOPA for years now, and of course he has a huge foundation and funnels a lot of money towards research. And I think that that's fantastic because then his name is getting the word out, and they've come a long way. And it's helped quite a bit. I mean, he's got a new TV show. I watched an episode. It wasn't that good, but I like him as an actor. He's a great voice actor. But you can still, he's pretty shaky in his voice, right? But he hasn't declined nearly as much as you would have expected if he hadn't been on L-DOPA. So dopamine is not just about the reward pleasure center. It has to do with motor movements. It has to do with our sleep-wake cycle as well, arousal. We are steadily, all of us with normal firing, healthy functioning brains, meaning that you don't have some kind of chemical imbalance, either from something that you inherited or from doing drugs, right? You have a normal, healthy brain. We are steadily releasing dopamine throughout the day. And then you'll get spikes of it when you have either an enjoyable experience, or if you exercise, or you're with a friend, and you see these elevations of it. But you should be having steady streams throughout the day. You're awake, you're alert, right? You have purpose in life. That's all tied in with dopamine. Okay, whoops. Okay, so then endorphins, I already spoke about that a little bit. There are natural painkillers. Endorphin means opiate-like. Okay, so it's kind of like being high. The natural high, and this guy's on a runner's high. Of course, you have to be in good physical condition to feel the runner's high. Once you get there, you will never want to go back. You want to just keep running. Or like in my case, I'm a swimmer. I just want to keep swimming. I swim when I'm on vacation. I seek out places to swim. If I can't swim in an ocean, if it's too cold, I'll swim in a pool. I'll drive places so that I don't get out of shape, because going back home after being off for a week, and it's painful, right? So endorphins serve a purpose to block pain, but then they boost all of these other systems in our body. Endorphins then are going to boost your dopamine system. Okay. Serotonin, we tend to also think of serotonin as being a happy chemical, but I chose this picture of this woman who looks calm. She's relaxed, she's satisfied, she's drinking her tea. Serotonin does have to do with alertness, it does have to do with sleep-wake, but it's really more about this sense of well-being, okay, more so than like this any kind of elation, right? It's a sense of well-being, serotonin. So we like serotonin. We want serotonin in our system. Okay, and then lastly, norepinephrine. Now, norepinephrine works in concert with your adrenal glands, so your brain and your adrenal glands and your nervous system. And it really jolts your system, it wakes you up, and the purposes of it really are to say, hey, pay attention, something dangerous could be happening here. So it's going to open up your blood vessels, your heart rate is going to accelerate, your breathing is going to get faster, your pupils will dilate. It's causing all of these changes in your nervous system that probably from evolution were put in place or it evolved to say, hey, pay attention, you're up too high, you could fall. Or that could be a poisonous snake, back away. So norepinephrine is released in our fight or flight system, and we'll talk about that in our next class. But then this woman's jumping out of an airplane, and she looks like she's really enjoying it. So her perception, she's getting a norepinephrine release, and we might call it an adrenaline rush, right? Adrenaline and noradrenaline, they're released from your adrenal glands, but really what's happening is it's opening up all those blood vessels in her system, and then she's getting pleasure from that. So this comes down to kind of personality. Some people thrive off of these types of feelings. Other people, no way, I don't like that feeling of my heart rapidly accelerating. The thing is that if she does this over and over and over again, it'll stop having the norepinephrine release because her brain, she's smart, she'll realize that she's not really in danger, and so then she'll have to go and do the next most extreme exciting thing to release the norepinephrine. All right, we'll